now with Soufiane Bel Rhali. So he is our PhD student, and his PhD thesis mainly focuses on the connection between the gut and the brain, and how dogs can be models for that. So he will present our work on the link between gut microbiome and family dogs' behavior. So please welcome Soufiane Bel Rhali. And gut microbiome composition is associated with age and memory performance in pet dogs.
Thank you. Let's start sharing. Okay, thank you so much, Cecilia, for your nice presentation. It's really a pleasure to be here and present our work. So as you said, my main area of interest is the gut-brain axis, which is bidirectional communication between the gut and the brain. And my presentation title is going to be the gut microbiome composition, which is associated with age and memory performance in pet dogs, as our last publication showed. Let's start. So it is really important now to know that microbes are everywhere, in your phone, in your water bottle, in your hand, before and after you wash them, and literally everywhere. Our body is inhabited by trillions of microbes, which contain, at least as human, we have like many microbiome cells as human cells, and maybe even more. So this fact made us more microbiome than human, as you can see, figuratively speaking, of course. So in general, there are 5 bacteria phyla that colonize our body, including actinobacteria, as you see in the figure, bacteroidetes, firmicutes, fusobacteria, and protobacteria. This is the 5 major phyla, and their proportion differ across our body, as you see also here. So most importantly, microbiome live inside us, which is really important for us because it's considered as a unique, like a fingerprint. And they are very crucial for our health because they help us for digest food, regulate immune system, protect us against bacteria that cause diseases, even they are competing with each other, and produce vitamins such as vitamin B12. So and also there are many factors that affect our gut microbiome, including nutrition, lifestyle that individual adopted, uses or not of antibiotic that could lead to greatest impact on our mental health and aging process. So now scientists start thinking of humans as complex ecosystems colonized by different microbiome species who collaborate, as I said, and compete depending on the circumstances. So before moving on, we need to understand what is gut-brain axis. So I'm going to give you like a simple explanation. So gut-brain axis consists of bidirectional communication between our gut and our brain. And recent advances in research have reported that bidirectional communication between the gut and the brain could be done by neuron, immune system, and hormonal links, like for example, neurotransmitter, as you see here, dopamine, cortisol, other hormones. And for instance, the microbiome in our gut here produce a whole bunch of different molecules. For example, we can see here serotonin. It is critical for as it is a messenger in our brain, and it is especially one known to influence our mood. This is why a lot of drugs prescribed for depression and anxiety affect serotonin signaling in the brain. But recently, it turns out that the majority of our body serotonin produces in our gut, which is a really great finding. Now we can just change the microbiome community there and just introduce the microbiome that produces serotonin. With that, we can manipulate our serotonin level in our blood and also manipulate our behavior. So the long story short here, this interaction is so complicated, and we are only on the edge of this picture, and more mysterious now finding challenge our understanding. So hopefully in the future, great discoveries are expected to shed light and reveal the truth behind this field. So before moving forward, we need to establish really good funding because the aim of the research is to translate and compare our results that we have found and we will get in the future with human findings. So a recent breakthrough which changed our understanding to the model used for like this interaction, the gut-brain axis. It came on 2018 by a group of research led by Luis Pedro Coelho from European Molecular Biology Laboratory, which is very known in Europe, reported that dogs are more similar to human in gene content, of course. As you see in the figure, according to their analysis, they had been done to compare gut microbiome of human genes to pigs, mice, and dogs. The results showed that there is an overlapping similarities between dogs and humans in terms, as I said, in gene content. This means that dog will be our next model in the gut-brain axis studies instead of pigs and mice that over years indicate they have been used over and over again to confirm this interaction without knowing that we don't have any similarities between pigs or mice. Pigs, we have like some assumption, as Tamsha has drawn our attention to it. We need to do more research in order to see that pigs that are living now in families that could have some similarities as dogs. And here, so after we have seen that really there is an interaction, there is a connection between the gut and the brain. So the real question here is, can we see this kind of interaction in dogs? So this question has been answered by Nico from Oregon State University, who showed that the gut microbiome really is linked to dog aggression. And most importantly, they have indicated that the gut microbiome may be useful for diagnosis, aggression, behavior, pre-order manifestation, which is a really great finding. It's like having a blood analysis that you are close to getting diabetes, so you can just make like some measure to avoid this. In this research, they have identified some bacteria species which are really correlated with dogs' aggression behavior. In this case, it is very feasible to alter and change the gut microbiome in this regard. So far, we have more questions regarding the dog's gut microbiome that we have addressed in our research. Can dogs be similar to humans in this regard as well? For example, is the relationship between aging, brain function, and intestinal microbiome similar to humans? So based on this question, we have investigated the relationship between the gut microbiome and dogs' memory, performance, and age. And we have worked on 29 dogs from various breeds. The experiment that we have done, we have run like behavior tests consists of memory tests. This memory test consists of 5 open containers, as you see here, were positioned at equal distance from each other in semicircular arrangement. Let's see a video to have like clear visualization how we done this test. So on the starting position, the dog witnessed the baited container that the experiment going to make the food in the container. So with that, both the dog and the owner, and alongside with the experiment, is going to work out from the room for like a period of time, like 30 seconds. So after this 30 seconds, the dog is going to be returned to the room and is going to be literally to find the baited container. So the task was repeated 5 times, and each container was baited once in a random order. Okay. After like behavior test, feces were collected. The most important point that we would like to draw your attention to it, this is the duration between the collection and the storage. That must be within 15 minutes, which is really crucial for our research, since poop is considered as a snapshot of the gut microbiome communities of your dog. So the question here that we have received, why 15 minutes and not 1 hour? The answer is very simple. Bacteria don't grow and multiply the same way as animals or humans. The bacteria can double every 20 minutes. One bacteria can multiply for 60 million within like 8 hours and 1 billion in 10 hours. So controlling and respecting the storage condition and the time during the collection and put the poop into the freezer is really important to evaluate the gut microbiome communities in precise manner. This is really important in this research. Here, like the microbiology part that we are using in microbiology department is like the workflow that we are using. It starts by collecting, as we have just seen, the samples, then proceeding with extracting the DNA from those samples. After that, we go for amplifying the target genes for multiple copies by PCR. Then we check the quality of the PCR experiment by the electrophoresis technique. Then after that, we made like DNA sequencing techniques. And the final part is the bioinformatics, where we clean the raw data and identify the species that can be more abundant in the samples. So let's see the result. So after undergoing bacterial identification, the results show that 4 major phyla in the dogs were Bacteroidetes, Firmicutes, Fusobacteria, and Actinobacteria, which is and also the genera that we have detected here, which is Bacteroidetes, Protovella, and Fusobacterium. Those bacteria, phyla, and genera was detected in the previous study, and they are considered now as the major bacteria on the dog's gut. The experiment that we have done, our first experiment, is to see the relationship between Fusobacteria and dog's age and general gut microbiome. We have seen that Fusobacteria decrease as dog gets older, and we have compared our result with the previous studies that have been done in dogs. We can see that this bacteria was reported with high dogs that have more access to outdoors. And this is the first time that Fusobacteria is linked to dogs aging. And also, this is really important to know which type of bacteria, its abundance fluctuating with age, because it would help us to develop a potential probiotic, as a case of human. Bifidobacterium is the species that fluctuate with age, and now all food industries like Nestle, Danone, targeting this bacterium as a potential probiotics. The second analyzing that we have done is we have analyzed the relationship between the actinobacteria and bacterial abundance and the number of mistakes during the memory test. As you see here in the figure, we see that the result showed that the abundance of those bacteria, the bacteroidetes and actinobacteria, increased as the memory mistakes increased. So we compare our result with the human findings. We found a similar association in the study that had been performed by Nicholas and his colleague at the Wilkinson Alzheimer's Diseases Research Center, who reported that those kind of bacteria, which is actinobacteria, alongside the bacterioids population that was associated with patients with Alzheimer's disease, which is a really great finding, which has a similarity with dogs finding. And also a recent study that has been just published a few months ago, I guess in November or December, they have they have analyzed the blood of the Alzheimer's patients, and they have found like some specific compound called liposaccharide, which is one of the major constituents of bacteria. And from this point, we can have like clear cut that, okay, there is a connection between the gut microbiome and microbiome in general and the diseases of Alzheimer's. As we said, we are on the edge of this big field, and more research are needed to confirm this findings. The third analysis that we have done is to see similarities between symbols. We have done what we call numeric dimensional scaling, it's called NMDS ordination, and the results show that the symbols are more similar to each other. Specifically, bacterioids, Fusobacteria are more abundant across all symbols. In addition, we have detected Lactobacillus genus and Faecolobacterium, which is two kinds of bacteria that's capable of producing short-chain fatty acid, specifically butyrate, which is really, really important for brain development. So as a conclusion, we can say that our gut-brain axis research started and directed under supervision of Kubinyi. We have found that there is a correlation between gut microbiome and memory performance, and dogs as well as we have found which kind of bacteria its abundance fluctuated with age. This is really, really important to see what kind of bacteria their abundance decreased with age, so that we're gonna target this for like potential probiotic for dogs. However, as we see here, the correlation, we have to make a distinction, because the correlation is not causation. And this is the highest importance when it comes to gut microbiota. Once we have now established, okay, there is a correlation between memory performance and dogs and gut microbiome. So now more research, well-designed experiments are needed to figure out what causing what. Okay, this bacteria A could cause Alzheimer's diseases. This is the most important point in order to prevent, or may say to cure, Alzheimer's diseases, even though some publications have said that, okay, Alzheimer's disease is irreversible, but who knows, because this gut microbiome has broadly effects on our body. So from there, we can develop new therapies that address the root of cause of diseases. This is our first published gut-brain axis research or paper that targets an animal family dogs. And have been published in Animals Journal, and we were very lucky that our paper has been selected over 300 papers and issue cover, as you see here. So I would like to give you a take-home message that the project that we are working on and is only for the objective to explore and describe the microbiome in family dogs, which can help us to understand not only similarities to human, but also the important differences, because identifying such a differences allow us to provide our beloved pets with the best care for their microbiome requirements. Thank you so much for your attention and your attendance, which is really, really appreciated, and for coming to our department to run this experiment in order to, for the sake of dogs and for our sake as a human, because it is a really great help that you are providing to us. Many thanks. Thank you.
Thank you very much, Soufiane. We have one question from Amanda. So, could there also be a common cause for differences in microbiota and memory, for example, stress?
As you said, gut microbiome is so complicated because also stress could affect the gut microbiome. Stress also have some effect like producing some, it's like enhance producing some hormones like cortisol that can also affect our capacity, cognitive capacity. But now we can see that the gut microbiome is also affected by stress and also can produce more hormones and more like precursor, like messengers, and also other compounds that can affect our behavior and our compounds.
Great. Next question is from Jackie. So Jackie, sorry.
Yeah.
Can dietary probiotics or food ingredients then be used to target amendments in the microbiome for supporting healthy aging?
Yeah, probiotics, as I said, is like a tricky and sketchy word, because probiotic now, as I said, now as I said, as I showed you in the first analysis that we have done, that some kind of bacteria called Fusobacteria has decreased as dog gets older. It is really important to study this kind of bacteria to see, okay, we can just address this bacteria to dogs, because Bifidobacterium or Lactobacillus is not probiotic for dogs, as most of industry is addressing this to the dog's owner, because Lactobacillus is not abundant in dogs' gut. It is like minor, it has like minor fraction in the dog's gut, and also Bifidobacterium, it's approximately doesn't exist, only at like from 1 month to 6 months, that's all. So as I said, Fusobacterium that we are now targeting, that we have shown in our experiment and our result, it's really important to develop probiotics. And before develop probiotic that targeting this bacteria, we need just to see what metabolic that this Fusobacteria are producing. Yes, according to your answer, if we have like probiotic, we can just support our dog's healthy aging. And we are like predicting this kind of bacteria is really good for our dogs to have like better aging in the future.
Great, very interesting and exciting news for the health and the good behavior of dogs. So thank you very much.
Thank you so much.